Okay, class, good day. So, for today's lesson, class, we will tackle your Module 5, which will be all about your antigens. So, when people get sick, class, it means that their body is being invaded by harmful substances that would cause an immune response in the body. Now, by identifying the said pathogens in or markers on cells, the body would then produce antibodies to fight these pathogens or harmful substances, and they would try to eliminate them. These invading microbes class are called your antigens. So we will tackle this further class, the so-called antigens. That are the ones responsible class for harming our body and creating antibodies. Now, what are antigens class? Now, please remember, as mentioned in the previous module, your antigens class are substances recognized as foreign by the body and have the capability to react with a complementary body, antibody, or a cell receptor. So let me repeat class in a simplified manner. Antigens are any type of foreign, it could be a foreign substance, and it's capable to cause class the creation of complementary. When you say complementary class, they would have a pair or a specific target. So they are the one responsible for the creation of complementary antibodies. So that's your antigen. Now let's talk about the different types of antigens class. The first one would be the autoantigen. Now when you say auto, from the prefix auto, this would refer to self. So these are self antigens. These are antigens class that would belong to the host and they do not evoke an immune response under normal circumstances. Now, I want to emphasize here, class, under normal circumstances. Now, during times, class, when an individual could be suffering from disease, this autoantigens would cause what you call your autoimmune disorders. Now, this would be tackled in your finals, but this autoimmune disorders class, one of the most common example would be your SLE, your systemic lupus erythematosus. Now, in the previous academic year, I had a student in Immunocero who also had this type of disease. She was around 21 when she first had her lupus. Now, according to her immunologist, to her doctors, the reason for the possible cause of his lupus was stress. So this is one of the environmental factors class that could trigger an autoimmune disorder, stress. So as students, please always take care of yourself, of your body by eating a healthy meal and drinking your vitamins and trying to get the next type of antigen would be your alloantigens. Now, this would come from other members of the host species. Now, one of the most common example of alloantigens in humans class is the blood group system. And one of the most common blood group system is your ABO blood group. Now, to explain this, let me just go to this slide. Now, ABO blood group system and their corresponding antigens and antibodies. Now, when we deal with your ABO blood group system, we need to understand that this is related to your blood type. But before we tackle this, I want you to take note of two things that you need to remember about antigens related to your blood group system. The first one, class, is that your antigens, your antigens for the blood group system are found in your red blood cell. The second one, the antibodies related to your blood group system. Please remember this. The antibodies are found in the plasma. Now, I want you to take note that when there would be a blood transfusion class, when there would be a blood transfusion, the thing you need to remember about blood transfusion, when it comes to the patient class, the thing you need to take note of is the antibodies found in their plasma. If you're the donor class, if you're the donor, the thing you need to remember or to take note of is the antigen found in the red blood cell. So let me repeat, whenever you're going to perform blood transfusion class, this is related to your alloantigen. The antibody should be found in the plasma is for the patient, while for the donor, you need to take note of the antigen found in the RBC. Now, let's go to your blood type. So, for you to understand what are alloantigens. Now, for your blood type class, there are four known blood types: A, B, AB, and O. So, this is the ABO blood group system. Now, for every blood type class, there is a corresponding antigen and antibody. Now, their relationship class. Their relationship is opposite or inverse. If you are blood type A class, if you are blood type A, please take note that you have the A antigen in your red blood cell. If you are blood type A, let me repeat, you have the A antigen in your red blood cell. If you are blood type B, you have the B antigen in your red blood cell. If you're blood type AB class, you have the should be A antigen and B antigen found in your red blood cell. If you are blood type O class, you neither have A or B, nor A, A nor B antigen. So you have no A or and B antigen, so no antigen. Now, as I mentioned, they would have a corresponding antibody and they would have an opposite or inverse relationship. So if you're blood type A, you would have the anti-B antibody. For blood type B, you would have the anti-A antibody. For blood type AB, since it both has A and B antigen, it has no antibody. And for blood type O class, since there is no antigen, it has anti-A and anti-B antibody. Now, these are the basic principles or the rules about your blood typing. Now, let's talk about your cross matching because your alloantigens are important in blood transfusion, specifically for cross matching. Now, let's say, class, your patient would require blood transfusion. He needs blood to survive. Now, the patient class is blood type A. The patient is blood type A. The donor class would be blood type B. Now, as per the rule, if you are the patient, as I mentioned earlier, the thing you need to remember about the patient is the antibody present in their plasma. In their plasma. Since it's patient A class, it would have the anti-B antibody and it would have the A antigen. While for the donor, as I mentioned earlier, the thing you need to remember about donor is the antigen found on their RBC. So it would have the B antigen and it would have the anti-A antibody. Now, if the patient A class receives blood from donor B, what happens is that the B antigen found in the donor red blood cell. Let me repeat, the B antigen found in the donor red blood cell will be attacked class by the anti-B antibody. And this would cause incompatible blood transfusion. And if it's incompatible class, it would lead to hemolytic transfusion reaction, where the patient would suffer from hematuria, hemoglobinuria, and other diseases and other symptoms that are capable of causing death. So let's do another example for you to again remember, for you to better understand this. Let's say this time class, your donor or your patient would be blood type B. The patient is blood type B, while the donor this time would be blood type O. Now, the thing you need to remember again, if we're talking about the patient, is the antibody present in their plasma. So if it's the blood type B, it would have your anti-A antibody, and it would have the B antigen. But if you're donor, when we're talking about donor, the thing you need to remember about donors is the antigen. So they should be compatible, or there should be no antibody that would attack the antigen and such. So if it's blood type O, as mentioned, it has no antigen, but it would have anti-A and anti-B. Now, what happens here is that since your blood type O, the donor, has no antigen class, it has no antigen, no antibody, no antigen rather, will be attacked by the anti-A antibody found in the B antigen. Therefore, this would be compatible. That is why class, your blood type O, your blood type O class is considered as the universal donor blood type. So it could be transfused to anybody. And let's do the last example. So I hope you're able to get this topic. This will further be discussed in your immunohema. But just to give you an idea, class, what would happen if you mix the improper alloantigens? It could be deadly. So this time, let's say this time, class, that your patient is blood type AB. Now the donor this time, class, is blood type B. Again, if we're talking about the patient, the thing that needs to be compatible with should be the antibody found in the plasma. So the antibody shouldn't be able to attack any antigen. So if it's AB, the blood type is AB, there would be no antibodies, but it would have the A antigen and the B antigen. If it's donor, again, if you're talking about the donor class, the thing you need to remember here, it should be that the antigen should not be attacked. So it has the B antigen and the anti-A antibody. Now, what happens here, class, is that since there is no antibody found in AB positive, AB blood type, there's no antibody found in your AB blood type, the donor B antigen would not be attacked, and therefore this blood type is considered compatible. It could be transfused. That is why, class, your blood type AB is considered the universal recipient. It could receive any type of blood. So I hope that was clear. The alloantigen, the most common type of alloantigen, the ABO blood group system. So let's continue. So those are your alloantigens. Next class would be your heteroantigens. These are antigens from other species, such as your animals, plants, and microorganisms. Now, to simplify this class, heteroantigens class are your bacteria, your viruses, and your fungi. While your heterophile antigens are heteroantigens that exist in unrelated plants or animals. Now, when you say unrelated plants or animals, they are separate or they would have a different species, but are either identical or closely related in structure so that antibody to one will cross react with antigen of another. So, here's an example of this. Let's say class here would be your salmonella and your shigella. So, this would be your salmonella bacteria. This would be your shigella bacteria. Now, as mentioned here, they would have an identical or closely related structure or epitope. So they have the same structure, the same appearance of the epitope. So parehong square, kunwari lang class. This is just an example. Now, when your salmonella class causes the creation of antibody, since they have the same structure of antigenic determinant site, the antibody created by salmonella can cross react. It can cross react with the antigen Shigella. So let me repeat: your heterophile antigens are antigens that exist in unrelated plants or animals, meaning they have separate species that have either identical or closely related structure. So when your Salmonella and Shigella would have similar epitope or antigenic structure, when your Salmonella causes the creation of antibody, the antibody created by Salmonella class could cross-react with your Shigella. So that's your heterophile antigen. Then we have also your homologous antigen. So this is an antigen class that induces an antibody and would react specifically with it. So it's very easy to understand class. This is just a specific. It has a specific antigen to antibody reaction. So if it produces antibody due to salmonella, it would target salmonella only. Heterologous antigen naman class would refer to an antigen that would react with an antibody it did not induce. So this is a type of cross reaction. And immunogen class are antigens which are able to elicit antibody mediated response in a responsive host. So these are the different types of antigen. So let's continue. Here's a review question class. ABO blood group system belongs to what type of antigen? Now let's talk about your immunogen versus antigen class. Now antigen class would refer to a substance that would react with an antibody or sensitized T cell but may not be able to evoke an immune response. Well, your immunogen class is any antigen that is capable of inducing humoral or cell-mediated immunity. This ability is called immunogenicity. Only an immunogen class can invoke an immune response. Thus, all immunogens class are antigens, but not all antigens are immunogen. To simplify this class, not all antigens can cause the creation of antibodies, while your immunogen are the antigens capable of creating antibodies. So here's the place that all immunogens are antigens, but not all antigens are immunogens. Let's continue. Now, factors influencing your immune response. So age. Now, the thing you need to remember about age class is for newborns class. For newborns class, when they are typically born, they don't have antibodies. It would take a couple of weeks for them to develop their own antibodies. That is why, for newborns to survive, they need to have a passive transfer of antibodies via the colostrum or via placental transplant while in the uterus. So that's for age. And another thing would be the older you get, the greater your age, the weaker your immune response, which is common among elderly. Then we have your health class. So if you're suffering from stress, you're suffering from uh, malnutrition, all of this class could cause a weak immune response. But if you have a proper diet, you're able to relax, then you would have a good immune response. Then for the dose class, so the greater the antigenic dose, the greater the immune response. So if it's a very large amount of antigens class, it could cause a very active immune response. Then routes of inoculation class, so this could be different routes of inoculation, such as your subcutaneous, intramuscular, intravenous, transdermal, implantation, and even oral administration. Now, the commonly used for your vaccine are these two. So it could be IM or subcutaneous, and they tend to produce better immune response. Then there's also your genetics. So in genetics class, in the finals class, we will talk about different primary immunodeficiencies. So there are some genetic diseases. When you say genetic diseases, they are inherited. So we will tackle the inherited disorders that could affect the immune system. So that's the factor. So age, health, dose, routes of inoculation, and genetics. Let's go to the characteristic of antigens class. The thing you need to remember of your antigens is that the higher the molecular weight, if greater than 10,000 Daltons, the better they are able to cause an immune response. Then chemical composition and molecular complexity. Proteins are the most immunogenic, followed by carbohydrates, lipids, and nucleic acid. So if you're going to arrange them from the one that's the most capable of causing an immune reaction, it would be proteins, followed by carbohydrates, followed by lipids, and nucleic acid. Then the more complex a molecule, the more immunogenic it is. Then foreignness. It must be recognized by the body as non-self. An ability to be processed and presented with MHC molecules. So these are the characteristics you need to take note. The ability to be processed and presented with MHC molecule class is a mandatory to become an effective antigen. Let's go to the nature of your epitopes. Now, epitopes class are known as your determinant site. As mentioned in the previous module, these are the ones where the antibodies Fab site would attach. There are also antigenic determinant of the antigens that bind to antibody or T cell receptor. So this is very explanatory class. I've already explained this since module 1. This is your epitopes, also known as your determinant site. They are found in your antigen. There, this one. Now, there are two types of epitopes class. The sequential or linear epitope. Your sequential or linear epitope are amino acid chains following one another on a single chain. Okay, for you to better understand this, this is your linear or your sequential epitope. So if you take a look at this picture, this is the antibody and it is already attached to the epitope. If you take a look at those beads, the one that are red in color, this one, these are known as your amino acid components of your epitope. Now, they would be forming a chain class that are arranged in a sequence. Meaning class, they are attached to one another and they would be continuous, just like this one. So that's the thing you need to remember about continuous or linear epitope. The amino acid change is in sequence and they are arranged. So that's your linear or sequential. Now, the other one is your conformational epitope. This would result from folding of one chain or multiple chain, bringing amino acids from different segments of a linear sequence or sequence into close proximity with each other so they can be recognized together. Okay. Regarding your conformational, this is also known as your discontinuous. Now, the difference between conformational from linear class is that when it comes to your conformational, the sequence class is not arranged. There would be spaces. So there would be spaces in between the amino acid sequences as seen in this picture. So this is caused by folding. So do remember the difference between the continuous and the conformation. Let's continue. Now let's talk about your haptens class. Now haptens class are non-immunogenic, meaning they are not able to cause an immune reaction. They would require a carrier class that would change its antigenic specificity, thus creating an antigenic determinant inducing an immune response. Now, for you to better understand this class, this is your haptens. Haptens class on their own won't be able to cause an immune response, but if you add hapten and a carrier molecule, an example of a carrier molecule would be your latex. The hapten would attach to the surface of the carrier molecule, and it would form your hapten carrier conjugate. Now, this one class is now able to induce an immune response. So let me repeat from the top. Haptens on their own class will not be able to cause an immune response. But if you include a carrier molecule such as your latex molecules, they would attach to the latex molecules, forming a hapten carrier conjugate, which will now be able to induce an immune response. Now, here's a review question. What is required for haptens to produce an immune response? Okay, now once antibody production is initiated, the hapten is capable of reaction with antibody even when the hapten is not complex to a carrier molecule. Now, when you say reaction with an antibody class, on their own, haptens could attach or could cause the attachment to antibodies. However, precipitation or agglutination reaction, when you say precipitation or agglutination reaction, this is the in vitro way. When you say in vitro, this is the one you would do in the laboratory class. In the in vitro way of identifying an immune response. So they would be via agglutination or precipitation. So if there is an agglutination, there is a precipitation, there would be immune response. So that's how we medtech identify if there's an immune reaction. So the positive result in most of your lab results sa immunosero would be positive for agglutination. If it's negative, no agglutination, something like that. So you will tackle that in the lab. Now, as mentioned here, precipitation or agglutinations would not occur because your hapten class only has a single determinant site and cannot form the cross links with more than one antibody molecule. Okay, in a typical antigen class, it would tend to have multiple determinant site. Now, with multiple determinant site class, it would cause multiple antibodies to attach with one another due to this multiple determinant site. So they would form a clump, they would cluster together, and that would cause a agglutination or precipitation. But in the case of your hapten, it only has a single determinant site. Since it only has a single determinant site class, it cannot form the so-called crosslinks with more than one antibody molecule. So imagine this would be another antibody. This would be another hapten. Let's say if this one attaches here, but since there is no determinant site here, it cannot form the crosslink, which is necessary for precipitation or agglutination. So hope that was clear. Let's continue. Now here are the characteristics of your hapten. Now antibody formation hapten alone cannot cause the creation. It cannot cause the creation. Please remember this. It cannot cause the creation class, but attach. It can attach to antibody on its own. It cannot cause the creation of antibody, but it can attach to antibody on its own. Now, hapten plus carrier class could cause the creation of antibody. So there would be antibody. Now here you have your hapten plus reaction with antibody. It would form a combination. Here hapten plus carrier. This one class. Hapten plus antibody. There would just be a combination. There would be no agglutination. There would be no precipitation. But if you include a hapten and a carrier, if you form a hapten and a carrier, so it would also attach to the antibody, there's a possibility class that it could cause agglutination, just like this. So the carrier is very necessary class if you want to induce agglutination. Now, adjuvants class. The power of immunogens to generate an immune response can be increased through the use of an adjuvant, a compound administered when an immunogen would increase the immune response. There are two common adjuvants class, your Front's complete adjuvant and aluminum salt. Now, an adjuvant is added to increase class the immunogenicity of weak antigens or enhance the speed and duration of an immune response. It acts by producing a local inflammatory response, which would attract a large number of immune system cells to the site of antigen exposure. It enhances the expression of co-stimulators and cytokines by antigen presenting cells. And these are the common components of your vaccine. So there are active components. It could be the antigen, should be virus, bacteria, or a toxin. Then the adjuvant, which would enhance the immune response to vaccines such as your aluminum. Antibiotics to prevent bacterial contamination such as your neomycin, gentamicin, and polymycin B. Preservative class would prevent contamination such as for repeated puncture of a multi-dose vaccine vial. Magnesium and thymrosal are preservatives. Then for stabilizer class, they would protect the vaccine from adverse conditions such as the freeze-drying process. They would include your sugar, lactose sucrose, amino acid glycine, and the protein gelatin albumin. Then there would be trace components class. So these are the leftover products from vaccine production, such as your formaldehyde. Then we have your super antigens class. So you're already familiar with this. If you still remember in your bacteriology, an example of a super antigen is your TSST, your toxic shock syndrome toxin produced by your S. aureus. So later we'll explain this. Now, super antigens class are a class of antigens that would result in excessive activation of the immune system. Specifically, it would cause non-specific activation of T cell, resulting in a polyclonal T cell activation and massive cytokine release. They would interfere with the adaptive immune system. So for you to better understand this class, I have to explain to you what would be a normal antigen presentation. Now it all begin with your antigen presenting cell. Now when we talk about antigen presenting cell, we refer here to the phagocyte. If you still remember in your phagocytosis, the last stage of phagocytosis is wherein the molecules from the remains of the phagocytized antigen would be presented with the MHC molecule. What happens here, class, is that the MHC molecule would present the antigen remains or the antigen molecule to the T cell via the T cell receptor, and it would interact with your variable alpha and variable beta region. Now, when it interacts with your both variable and variable alpha and beta, it would now cause a specific immune response. It would now identify, the T cell would now identify the antigen as a specific immune response, and it would release cytokines, and it would cause the movement of your WBCs to target the antigen to the site of injury. But in the case of super antigen class, they would bind first to the MHC class 2 and coordinate class only to the variable beta region. Now, when this happens, class, it would cause non-specific activation. When you say non-specific activation, it could target other cells, resulting, class, in a polyclonal T cell activation. When you say polyclonal T cell activation, the T cell will have multiple targets. And since it is thinking that it has multiple targets, class, it can cause a massive cytokine release, causing a cytokine storm. So that's how your APC would deal with super antigens. They would cause this massive cytokine release. Instead of a specific immune response, it causes a non-specific activation of T cell and polyclonal T cell activation. Examples of your well-known superantigens are your TSST1 produced by S. aureus, your streptococcal pyrogenic exotoxin produced by your S. pyogenes, and your staphylococcal enterotoxin produced by S. aureus, and your ETEC enterotoxin produced by your enterotoxigenic E. coli. That ends your module 5.